Good morning, everyone. I'm Jeremy Pickens with Alabama Extension, and I'm co-hosting this webinar. It's a collaborative effort between Alabama Nursery and Landscape Association and Alabama Extension. And today's guest is Dr. David Held. He's the department chair of the Department of Entomology and Plant Pathology at Auburn. And he and his student Ariana are Littler is going to present some research updates. I'm going to pass it over to David and let him introduce the project and Ariana.
Yeah, thanks, Jeremy. So don't give me all the credit. Jeremy is a co-advisor for Ariana Littler, who's a PhD student working with us on this Chilli Thrips project. So we're about 2 years into this project, and I think we've got some really good information to share, not just on sort of when we can now expect to see Chilli Thrips, but some information on biological control and resistance management, which are some really important issues. So with that, I want to just kind of turn it over to Ariana to talk about her research over the last couple of years and give you some really good takeaway points to walk away from this webinar with. Ariana, take it away.
Okay, good morning, everybody. If I can get my computer there we go. Okay, so Chilli Thrips are a widely invasive insect native to Asia and the Indian subcontinent, and officially came into Alabama around 2017. And we determined that they're up in Auburn as of this past November. So they're spreading pretty quickly. Not only that, they are a species complex, so they have a lot of individual species within this species that we can only tell with DNA barcoding. The strain that we believe is present in Alabama is the South Asia strain 1. And unfortunately, there is many instances of developed resistance to insecticides, both in the native range and even more recently in Florida. As of last spring, I believe, there was a paper that had come out showing that they had increased resistance to a lot of commonly used insecticides. And bifenthrin, for example, they had no more than 60% mortality when they were trying to treat Chilli Thrips with bifenthrin. And so we're trying to figure out a new resource to mitigate insecticide resistance, whether that is using different types of chemical controls or even biological controls. And this image, I think, says it pretty well how this species impacts the industry. It feels like a bull's horn going up where the sun don't shine for the nursery folk. So it does cause problems pretty quickly, and unfortunately, pretty decent problems. And these thrips are very small in comparison to other species. So in the image on the right is a Chilli Thrips compared to a Western flower thrips. You can see that they're pretty small, less than 1 millimeter. And they also have very short mouth parts. So unlike aphids that can pierce in and suck up the plant juices and just completely collapse the leaf, thrips have very small mouth parts and can only feed on individual cells. So they suck out the contents of those cells and essentially kill the plant cells. And that's what creates that stippling that is commonly associated with thrips. And they also have a saw-like ovipositor, and they cut into plant tissues using that ovipositor to lay their egg there. And overall, the egg to adult life cycle usually occurs in about 11 to 21 days. And that can vary depending on temperature. So they can live anywhere from 10 to 44 days after they reach adulthood. That 44-day span occurs at around 55 degrees Fahrenheit. And their egg production is also higher at those milder temperatures, with roughly 50 eggs per female in their lifetime, around that 70 degrees temperature. And they are highly prolificous, so they feed on many plant species within a large number of families of plants. They also develop the fastest on rose and also pepper, but for the sake of this presentation, we'll just focus on the rose. But you can see what the damage looks like in this image on the right on a whole bunch of different plant species. You can see that bronzing, silvering, leaf curling, withering. It can also have premature bud and fruit drop. And unfortunately, they do prefer that newer growth due to oviposition and even potentially feeding, because it is softer and more easily punctured both by their mouthparts and their ovipositor. And forcing that new growth using fertilization and pruning, or fertilizing and pruning, can unfortunately cause a large boom in numbers if you're hitting it in that time period where they're already pretty high in numbers. And they are pretty fast moving. So in the video on the right, you can see a thrips moving along the plant edge. I don't know if you can see my mouse, but you can see them moving. And their daily flight period usually occurs between 9 a.m. to 4 p.m. Based on prior work. And they don't typically disperse very far. So after 17 days of being placed on an initial plant, on strawberries is what this Panthea all did, they found that there is more damage and thrips numbers on that initial plant as compared to those that are up to even 4 feet away. So they tend to stay pretty localized unless plants are touching each other. And we wanted to see what the seasonal activity was beyond just that daily activity. So we decided to pair with a couple different nurseries around Mobile, and we put sticky cards in and outside of blocks of host plants. So in the image on the right, you can see 2 red circles. Those are showing 2 yellow sticky cards inside of the block. There are then 2 more outside of the block. And they are placed once every 2 weeks to capture that 11 to 21 day life cycle period. And then also between the hours of 9 a.m. to 4 p.m. so that we can get when they are most active and flying around. And this in and outside of the block, that'll tell us whether the numbers are higher inside of the block of host plants or outside, and whether they are moving in or out. And this is what we have so far. We're still working on some counts that we've collected earlier this year, but the blue line is the thrips numbers, or the Chilli Thrips numbers outside of the block. The green line is the Chilli Thrips numbers inside of the block. And this is kind of showing where they're at, what time of year their peaks are. And based on the data we are seeing, we are recommending that you sample for Chilli Thrips numbers to figure out when you should start your treatment regime around September and March. So once every 6 months, the beginning of March, beginning, middle, and the beginning roughly of September. That'll give an idea of what the numbers are. And this is roughly 2-ish weeks before that peak, both in the spring and the fall. And so our conclusion so far is that there very much are thrips in and around the nurseries, and that you should monitor for thrips on these susceptible hosts once every 6 months, being March and September. And so some commonly used insecticides, this we had gathered from a survey that we had done last summer. These are the 6 highest reported insecticides used to treat for Chilli Thrips when they have that problem, with spinosad and or conserve and hachi being the highest 2. And that's just what the growers had reported. Unfortunately, there is a problem with insecticide resistance, and that, unfortunately, like I said, that South Asia 1 biotype is prone to that insecticide resistance, and application methods do aid in that resistance. In this blast sprayer, you can see it's blowing the insecticides on these plants. However, it's very indiscriminate. So that blowing, there is extra particles being blown out the side of that cover right there. Not only that, the force of the blowing could blow the already resistant thrips into other plots that are nearby. So how we are applying what is currently working could aid in potentially resistance development in later years or even months, who knows. But okay, there we go. So we can mitigate resistance using IRAC. So these are the number letter combinations that are associated with the different insecticides. So ARIA is 29, Conserve is number 5, and then Hachi Hachi is 21A and 39. For the sake of us, it's 21A. But the numbers are associated with the mode of action, and we want to rotate between mode of action. So you don't want to follow Conserve with another number 5. So you'll want to change the numbers between your applications. Which brings us to a new product that we're currently putting together. And this is a trifold that will give you information about general Chilli Thrips life cycle, what their damage looks like, how small they are, as well as different insecticides, both their commercial name and their active ingredient, as well as the level of control. The opposite flip side of it being a set of what you should rotate with each common insecticide. So here's what all of those look like. There's also instructions for sampling methods, how to contact your extension agent for getting more resources, more information. And I think it would be very useful. It would give you something quick and easy that you can just stick in your back pocket, carry around with you, and reference when you need more information. And if you're interested in having this trifold, you can scan this lovely little Thrips QR code, and it'll take you to a form where you can give me your email, your shipping address, and how many copies of this you would like, and we can ship them on over to you. And with that email, I can update you about the shipping when it should arrive. So hopefully y'all are interested in that. But here we're going to look into the other management practices because of that potential resistance. So there is the timing of pruning. With those peak periods, you could prune around those so that that new flush of growth is not occurring during when those peak numbers are in the nursery. There's also removal of weeds. So like I said, they are highly prolificous, and unfortunately, weeds in and around the blocks could be potential hosts. So while they might not be ideal, they could serve as a shelter of sorts when they are not feeding on the plants themselves or laying eggs. Also, reflective sheets or mulch. So those have been shown to cause a bit of a confusion landing idea. So the thrips won't be able to properly locate and land on their host plants because of the reflection of those. These papers, however, are in Japanese, and I am not fluent, so I cannot fully read those beyond the abstract, but they do have indications, so that is definitely good. And finally, utilizing biological controls, one of which in the image on the left is a minute pirate bug, or Aureus, very effective in controlling thrip species in past research. And Emblicius cucumaris, or just Emblicius mites for the sake of this, have been previously utilized for thrips control in flower thrips. Other thrip species have been shown to have effective control. And in another paper out of Florida, in 2009, they showed that these amblyceus mice actually had high levels of control of Chilli Thrips. So they had less than 1 Chilli Thrips per plant terminal, which is lovely. And finally, they also did a combination of amblyceus mites and those minute pirate bugs that I was telling you about. And that combination of those two species was found to be more effective than just the mites alone. So we wanted to try that ourselves. So we had 4 replicates of this, 2 up here in Auburn and 2 down in Mobile, with 4 different plant species. So that Arthur's paper just used 1 plant species. They used strawberry, I believe. But we decided to use 4 commonly used, like common ornamental plant species. So we used rose, cleara, dystillium, and azalea. And we also wanted to do 4 treatments. So we did no predators, which is the control, that is just the thrips, mites only, aureus only, and then aureus and mites. So the 2 pictures on the right show a sachet that you attach either to trellises or branches of the plant close to the stem, and the seasoning bottles essentially of aureus species. So both of those are what we had used. The sachets are loaded with 250 functional colonies or individuals of these mites, and they can breed and live on the plants. And then the aureus bottle that we had purchased was 1,000 aureus in 1 bottle, which both of these that we had used were the smallest number essentially that we could buy. And that is what we loaded onto the plants. And we then took plant terminals. We took around 2 plant terminals each, or 2 rose buds for the roses as well, and wanted to get our mites from there. Now, the rosebuds stole all of the thrips from all of the other plants. So there are virtually no numbers on the other plants for Chilli Thrips, but they were very prevalent on rosebuds. So the rosebuds control they had thrips numbers, they had Chilli Thrips numbers throughout, but all of the treatments did start with those Chilli Thrips, and then they rapidly decreased after the pre-treatment. So we did a pre-treatment sample, then we added the predators, and then we sampled for 5 weeks after addition. And so that pre-treatment, we can see that there's thrips there. And for each of the treatments, all of them go down. They can roughly maintain control for over 5 weeks. And while it may look like very low numbers, like the 2.25 that is present with the mites only, that we need to keep in mind. We are only taking around 2 buds worth of plant tissues from these roses, and knockout roses, they have a lot of buds. So if you take this and think about how many buds are on a rose plant at once, that actually can show that there is a lot more going on than what appears in these graphs. And it looks that the aureus or the minute pirate bugs are doing a lot of the heavy lifting here. They maintain that control for the duration of those 5 weeks, whereas there were tiny little blips in the mites only and the mites and aureus together. But it is showing that there is levels of control here. And so just some conclusions. These predators are inexpensive. So they can equal, so the mites that we had ordered, we got 100 mini sachets of these 250 thrips. And that is about $28.95, so about 28 cents, 30 cents per sachet. And the aureus was about 111 for that 1,000. And that would be about 11 cents per mite. And we'll get into the the release rate in another slide coming up. But like I said, those thrips were mainly found on rose buds, and this can show actually that rose could be a potential trap crop, not crap crop. So it shows potential for rose as a trap crop. So you can place a couple roses nearby that are budding, and that should attract them away from those other host plants that you are trying to protect. And also they are effective at controlling thrips numbers over multiple weeks of time. And we wanted to see what the release rate was for these predatory mites. So like I said, we have those sachets, and there is, the way we set it up is we put them in on top of a Ziploc bag, surrounded them with sticky, and each week we sampled how many thrips were being released. This is a very short video, but you can kind of see those mites moving around for a second. So they're very small. You can't see them when they're on the plant. So it's not like they're going to be an eyesore. And let's switch slides. We had it set up in an incubator. So we did 3 different replicates of it and wanted to see how the numbers came out every week. So they did have continuous release for up to 4 weeks. It ended up being roughly 1,600 to 1,700 mites total. And keep in mind, like I said, they are preloaded with 250 mites. So that does confirm that it is a functional breeding colony. And the highest numbers were released within the first week. So it was anywhere between about 900 to about 1,200 released within that first week. And that functional breeding colony does have potential to sustain on plants if there are other ample resources. If there are ample prey, and also potentially pollen. They do feed on that when prey numbers are low. So that can help aid in sustaining your plant colonies. And the main takeaways with this presentation is essentially, we've got to monitor for those chili thrips every 6 months so that we can keep an eye on what our numbers are within our select nurseries. So that March and September, before that peak period, and we can treat from there. And then also insecticide rotation should be implemented by utilizing those IRAC numbers. And that's a huge thing so that we can prevent resistance development at all costs, because it'll be more expensive in the long run if they are resistant, unfortunately. And with the biological controls, like I said, they're active about 4 to 5 weeks after release, and they can be for hot spots or problem crops. So they can control those numbers. If you see plants that have an infestation, you can tell by the damage, then you just stick these guys on there. With the aureus, they come in that seasoning bottle, so you can just sprinkle them on the plants, season those plants a little bit, and then just stick those sachets on them. And so Green Methods is where we had gotten these predators from, and they recommend that it ends up being about 1 sachet per 10 square feet. So 100 sachets treats about 1,000 square feet. And then the 1,000 aureus, these are a lot more mobile than the mites would be. So they also say about 1 aureus per 10 square feet, which would then end up being about 1,000 to 2,000 square foot control. So that would be the 112 for that amount of square feet, or the $29 for that square feet. So it should be relatively cost effective as compared to other chemicals that could be more pricey for the same amount of space. And if you have any questions, please shoot them at me. If not, my email, and then the QR code again, if you're interested in that trifold.
Thanks, Ariana. The other thing I'll mention about the biological control costs is the offset potential of resistance management. So by adding just that other practice, it's just another tool we have available to us, especially with something like Aureus, to help offset and mitigate the development of resistance.
How do those, how do the Aureus handle the, let's say the spring and fall cooler months? Let's say you were trying to use them in an overwintering house. Would they, have similar performance, you think, in the winter?
If it's in an overwintering house, I would think so. Obviously outside insects are a lot less active and prevalent in the winter. But in a greenhouse or overwintering house, they should have pretty decent control. I'm not sure. I haven't personally looked at what their seasonal behavior is, but I have personally, when I have been collecting in the spring and the fall, I have seen active Aureus on the plants.
Cool. Have you seen any research or know of any trials that have used some of the beneficial nematodes? I don't know, isn't there like 2 instars of Chilli Thrips that were there on the top of the pots?
Yeah, I personally don't know. That would be interesting to look at. But yeah, the pupa can pupate within the soil or the top layer of the soil. So I could see the nematodes being beneficial as well. I'll have to look into that.
So Jeremy, back in 1,000 years ago, it seems, when I was a grower, I actually was using Steinernema nematodes in a greenhouse for Western flower thrips control. I felt like it was an important part of my IPM program when I was a greenhouse manager. Just another way to try and reduce my reliance solely on insecticides in the greenhouse. Not seen any data for Chilli Thrips. Deploying that outside might be a little more of a challenge. We had kind of a captive population in the greenhouse. So I would think if a greenhouse producer is watching this and they're having problems with Chilli Thrips, that would certainly be something to give a shot. I'm not really sure how to effectively deploy like Steinernema carpocapsae out in an outdoor production nursery.
Right.
Jeremy, would that be something that they could put in the potting line when they're potting plants up to add it?
I mean, yeah, yeah, I don't know how long, you would get the goodie out of it, but yeah, that could happen. I mean, there was a couple of nurseries down here playing with the, I guess, flowable nematodes. I think it's Nemesis, maybe. And, it was not a trial, but they put it out and they thought that it helped with red-headed speed beetle at a nursery. But so I mean, I don't know how they applied, if they put it through the air blast sprayer and just, cross their fingers or what. But they thought, there's no control there, but they thought it did some good for them.
They'll be fine going through like a hose on proportioner, dosatron type thing. That's pretty easy on them. If you start getting higher pressure, the nematodes will explode under higher pressure in the spray.
Okay,
Yeah.
I got you.
Yeah, so that could be an issue with releasing some of these nematodes through, depending on the amount of pressure in that sprayer.
Right. So I've seen some older stuff where they suggested some different rotations in that somebody mentioned a neonic as part of the rotation. Have you seen any efficacy data on any of the neonics or systemics?
So let's just go back to this.
Yeah, that trifold, we have a column that's expected control.
Yeah.
You look at something like a TriStar, that you get 2 to 3 weeks residual and pretty good control. I mean, that's a sprayable. And then Safari, same thing. So we are getting pretty good control with those materials. But the residual, again, we don't get a long residual out of most of these, which the other thing I thought was quite positive, the biological controls. We were getting a little bit longer residual than we would out of a typical spray rotation.
So if you were to put you on the spot here, but if somebody was Chili Thrips was a new pest for them, and you were to give them a recommendation, what would you tell them to do? How was the frequency, and what, let's say 3 chemicals could they rotate with just out of the gate?
That there's a couple that they could go with. I think conserve for sure, because that is one of the most effective at the moment. And then Hachi, and then I mean Pylon, I believe, is certified to greenhouse only, I believe. And oh, that's a great question.
I would add one of the systemics in there. I would probably at that point try to introduce either an Avid, which has translaminar systemicity, or one of the neonics into that rotation. So you are getting some plant uptake as well as just contact activity.
Yeah.
And what's the frequency? 10 days?
I believe that is.
I don't have all those labels memorized. I'm not gonna
Well,
go
I
on
mean,
and
you
do
know,
that.
I guess to
But I
break
think
the
usually
cycle.
it's a 2-week rotation, right? So if you look
Okay.
at most of what's been done and evaluated, they're trying with many of these to go every 2 weeks.
2
So I
weeks.
think the
Okay.
evaluations are about 2 weeks.
Yeah,
That's
I
why
think
there's the 2 to 3 week residual listed for most of these.
with the labels I've seen recently, there's a couple that were saying 7 to 10 days. And then I think primarily it was roughly 2 weeks for a lot of them. And that's if I'm recalling correctly.
Well, great. Is there any questions from the audience?
Got one. And you apologize, you may have covered this. It was a lot of information to absorb, especially for a non-grower like
Okay.
myself. But you certainly don't want to hurt your beneficial insects. So kind of like, how are you doing the release on or which chemicals are you using that's going to have the most minimal impact on the beneficials? And how do you coordinate that, I guess?
I'd have to, that's a hard thing, because if it's not a specific insecticide, so one of the more general ones that will cover multiple pest species can also get those beneficials as well. So you'd essentially have to, I'm not sure of this on the top of my head, but you'd have to find ones that are more specific to thrips species. So unfortunately, neonics have been shown to have problems with pollinators. So it can depend, really. I'd have to look at if there's anything that has shown how insecticides affect those beneficials, these specific ones also. But I'm not sure off the top of my head.
So
I didn't know if it
one of
was
the worst
like,
case scenarios, Russell, would be like Telstar. It's the most broadly toxic. I've evaluated against other natural enemies of other thrips species, and it nukes them. So I mean, if you're going to put Telstar out, you're going to pretty much lose all benefits from the natural enemies that might be out there. It's kind of the worst case scenario. And then if you move, Conserve was named Conserve because it came out with this green award when it was released as kind of a conserve approach for saving natural enemies, right? So conserve would be on the other end of the continuum, where it's relatively low toxicity to many beneficials. So somewhere along that, most of these others would drop. I just don't keep all that information on the top of my head, but that at least gives you some idea of the range there, from something that would absolutely clean the slate of all natural enemies like a Telstar, which, to exactly go back to Ariana's point, it's very broad spectrum, to something like Conserve or an Avid, which has more target specific or reduced risk, as we would consider, against natural enemies.
okay. I mean, I guess I was thinking along the lines of, not using a contact insecticide when the beneficials are out. Maybe you put a systemic out, and then a couple of days later, when it's absorbed and maybe washed off the leaves, then you release or something like that. And maybe y'all are doing that research, or that's on your list to look at.
Yeah, so one thing about the systemics with Aureus, aureus will actually drink a little plant sap. It doesn't cause damage, but it can
No.
drink a little plant sap to when prey numbers are low. And so they have seen in field crops where that can be an issue with certain systemics. That's the only concern, concern there.
I mean, also on that front, the master's student that was in our lab, she had seen that there was a mass die-off of beneficials when using a systemic because they were getting secondary doses from the prey they were feeding on as well.
Talking about conserve, that reminded me something I wasn't aware of until just this past year. Was a conserves label suggests a tank water pH. I can't remember what the pH is, but anyway, a lower pH in your tank water, your spray tank water, can affect the efficacy of that product because it's not buffered. So most of the wells, at least shallow water wells, and down here in South Alabama, have a pH, pretty low pH, down in the 5s, which might be, if you're not, there is resistance with spinosad or conserve, but that may also affect the efficacy of your conserve applications if your tank water pH is too low. Something you might want to check out.
Good point.
So based on what y'all are saying with those beneficials potentially dying off from the chemicals, I mean, is that the recommendation then? Is that the beneficial substitute for one of the 2 to 3 weeks of spray application?
Yes, and that it's basically start thinking of them as another rotation.
Any questions from the audience or? All right. Well, thank you guys for coming and joining us today, and appreciate this. And we'll have this recorded and up on the Nursery Association's website here in the next week or so. Thank you.